The fourth day of browsing genes and genomes with Ensembl workshop. My name is Alina Mushtaq and I'm in the Ensembl outreach team. Today I will be presenting Ensembl comparative genomics. I've pasted the training link in the chat, and this is the link to the Nextgenhelper training page where you can find the coursebook that has the exercises and also the PDF versions to the presentations and the living document, which has space for questions and some useful links. So let me just go into the presentation mode. It's a very exciting time for comparative genomics, as there are a lot of large consortia that are aiming to sequence all organisms. So for example, we have the Darwin Tree of Life, which aims to sequence all eukaryotes in British Isles. There's also Vertebrate Genomes Project, which aims to sequence all vertebrates on Earth, and Ensembl is heavily involved in both of those projects. The objective of comparative genomics is to examine evolutionary relationship and find differences between the genomes, but more importantly, more practical applications are to drive gene functions based on homology, so we can learn something about the gene function of a less studied organism, and we can learn and more obscure species by looking at its equivalent genes, so its homologous genes in better studied organisms, but also the other way around. We can learn about the gene function in humans by manipulation in model organisms, and all of the animal research is based on this assumption of homology. Comparative genomics is also used to identify highly conserved regions, which tends to be similar across taxa as they are highly functional, and any new variation is likely to be deleterious in those regions, is being removed by the purifying selection. So we perform comparative genomics analysis by taxa. We have vertebrate comparative genomics in Ensembl, which also includes a few non-vertebrate model organisms, which serve as an outgroup route to the tree. And then you can find comparative genomics analysis in Ensembl genomes done separately for plants, metazoa, fungi, protists. Unfortunately, there is no bacteria comparer as there are just too many of them. You can find comparative genomics analysis on the Gene tab, and this is a tab that we have explored previously this week. You will see on the menu on the left-hand side, there is a section dedicated to comparative genomics over here. So for example, here I'm showing you the example of plants. But also we have wider comparative genomics analysis across taxa, which is called pan-taxonomic compara. And you can see it also includes a selection of bacteria strains. It's not done for all species in Ensembl and Ensembl genomes, only for a subset of representative species across all divisions. And the pan-taxonomic compara can also be found in the Gene tab, but only in Ensembl genomes. So if you're working with vertebrates and you would like to have a look at this wider pan-taxonomic comparison, you can get there by a non-vertebrate ortholog of your gene of interest. So if you're working with a human gene, you first have to find the non-vertebrate ortholog, for example, in yeast, and through there you can get to this pan-taxonomic analysis. We have comparative analysis for more closely related species. For example, we have the mouse compara, which includes all of the mouse strains and species, but also rat, and we have similar comparison for pig breeds and other agricultural mammals. There are different comparative data types that you can find in Ensembl. So we can distinguish gene-based resources, which can be found in the Gene tab. We have phylogenetic trees, for example, protein trees and non-coding RNA trees, but also homologies inferred from the gene trees. And we also have stable ID mapping. And there are different comparative data types that you can find in Ensembl. So we have the second type of comparative analysis data you can find that are called sequence-based resources, which can be found in the location tab. And here we have analysis derived from whole genome alignment. So for example, the reconstructed ancestral sequences. We have base scores such as age of base, conservation scores, constrained elements, but also syntonies, which is a conservation of gene order on a chromosome. And we will look at syntonies a bit more in the demonstration. So what I have been calling gene trees are actually protein trees. And we don't calculate gene trees for all of the transcripts in Ensembl. So what we do is we take a representative set of translations and we use one translation per gene from all species, and the representative translation is called Ensembl canonical transcript. We then perform all versus all model search to classify those representative translations into families or clusters, and we then calculate a multi-protein alignment for those clusters. And from the alignment, we calculate the phylogenetic trees for each cluster, and we also perform reconciliation against NCBI taxonomy. And then finally, we also infer homologies from the trees, so we could infer orthologs and paralogs, and we have a similar pipeline for non-coding RNA trees. So there are different types of homology relationships. So here we have a simple tree, and all of these genes share evolutionary origin. And speciation events are shown in blue, whilst duplication are shown in red. So you can see that we have a duplication event in primates, and as a result, we have two copies of this gene in chimp and in human. And genes which emerge through speciation event are called orthologs. And there are different types of orthologies. There are one-to-one orthologs, for example, C1 in chimp and H1 in human, which is one-to-one relationship as there is one copy of this gene in each species. But then we can also talk about one-to-many orthologs. So for example, this M mouse gene has two homologs, two orthologs, so two equivalent genes in both human and chimp. So this M gene in mouse is homologous to both H2 in human, but also H1 in human. Finally, genes that are merged through duplication events are called paralogs. So for example, here we can see that C1 in chimp is paralogous to C2 in chimp. As you can see that they both diverge through this duplication event, and same goes for H1 in human and H2 in human. As we have two copies of each gene in both human and chimp, these are examples of within species paralogs. We can also distinguish between species paralogs. So for example, C1 in chimp will be paralogous to H2 in human, as those are defined by the event that caused them to diverge rather than the species. So you can see that C1 and H2 also diverge through duplication. We then have different representations of gene trees in Ensembl. So we have the standard gene tree, and then we have the gene loss tree, gain loss gene tree. And this is exactly the same data, but different representation of it. So this gene loss tree is kind of more collapsed, and it shows you the number of members in a given clade, while the regular tree gives you all of the information. So this gain loss tree kind of focuses on the gene contraction and loss event. Significant gene expansions are shown in red. So for example, here in human, you can see that the humans have 3 genes in this clade, while there is a significant contraction shown in mouse in green, which means there was a loss of gene in the mouse lineage. And now we're going to move on to the demonstration part. And this is the first demonstration. We're going to explore the gene-based resources first on the Ensembl browser. So you can find the demonstration on page number 56 of your course books, or you can just follow me along the screen as I go through it. So let me just jump out of my presentation. And I'm going to go to ensembl.org. And I'm going to search for human BRCA2 gene. So I'm going to specify my species. And I'm going to search for BRCA2. And this is not case specific. So now I'm going to click the first search result, which is BRCA2 human gene. And this lands me on the gene tab. So this is a tab that we have explored previously, but I would like to bring your focus to the comparative genomics section on the left-hand side. So under comparative genomics, we have genomic alignments, gene trees, gene gain loss tree, orthologs, paralogs, and ensemble protein families. I'm going to click on gene tree. So you can see that gene trees have their own ensemble IDs, starting with ENSGT. And here we have our gene of interest that is shown over here in the red text. So our gene of interest is in the context of the phylogenetic tree over here. And then if I scroll down a bit, we also have a really nice legend at the bottom. So let's look at the nodes first. So the tree tips or leaves are shown in white are these gene nodes. We then have the speciation nodes that are shown in blue. Then we have the duplication nodes as red. And then we have ambiguous nodes in cyan. And then we also have gene split event. We then have collapsed nodes over here that are shown in this gray, that are shown as like these gray funnels. And these are many related species of certain groups collapsed together. So for example, here we have bony fishes, then we have frogs and toads, reptiles and birds. These are collapsed by default, as otherwise the tree would be very big, but you can expand them by clicking on the node. So let's say that I would like to expand the marsupials. So I'm just going to click on this node. And this will open a pop-up box. Which gives me a lot of information about the number of homologs, the branch length, bootstrap, and also export options. And then we also have the option to view the subtree. Expand the subtree or expand all subtrees as well. So let's just jump out of this for a minute. Then we also have these protein alignments. And as I mentioned in my presentation, that the genes are calculated from protein alignments. And here we have a summary view of the protein alignments, so next to the tree. And this alignment is based on the representative protein for each species. So the green shading indicates alignment, while the white bits are gaps. And for the collapsed nodes, we can see a consensus alignment with 2 shades of green. So like this over here. So dark green means that above 2/3 of the collapsed sequence are aligned. Light green means that between 1/3 and 2/3 are aligned, and white indicates that less than 1/3 of the collapsed sequence are aligned. all of this is explained in detail in the legend below. So over here, you'll see collapsed alignments and expanded alignments section. So I'm going to go back to the marsupials and click on this node. And I'm going to click on expand this subtree. So as you can see that after expanding the marsupials, we can't see the dark green anymore. So we have the individual alignments shown instead of the collapsed alignment. And lastly, those black lines in the alignments indicate exon and intron boundaries, and they could be either conserved or consistent across the tree. And another thing that I wanted to show you is that if we also click on the node, we can also view the subtree. So we can view it in Wasabi Viewer, which is an external tool. And it's going to open a new window. And so it's going to show me the alignment of the nodes that we're interested in. And you can see that it's quite colorful and nice and pretty. So let's go back. And you also have the option to download data from this image. And you can download the data in different file formats. And you also have the option to guide to file formats, and you can preview the data or download it as compressed. We're now going to go back to the left-hand side menu, and we're going to look at homologous genes now. So over here we have orthologs and paralogs. So paralogs is grayed out. So it means that we don't really have paralogs information on it, but we're going to click on orthologs. So on this page, you're going to find two tables. So if I scroll down, you'll see that we have two tables. So the first table is the summary table where you can select taxa of interest, and it also gives you a summary overview of ortholog types across the different taxa. And the second table is all orthologous genes across all species in reference to our species of interest, and there are more than 200 of them. Sorry, around 167, not 200. So for every species, you can find the type of orthology and the link to gene tree. Then we also have information on the ortholog, so it links you out to the gene tab, and then we can also view sequence alignment. Then we also have the target percentage ID, which is the percentage of orthologous sequence matching to the human sequence. And we have the Curie percent ID, which is the percentage of the human sequence matching to the sequence of the ortholog. Then we have the gene order conservation scores, and these are from 0 to 100. So gene order conservation scores basically tells you how many of the 4 neighboring genes are also orthologous. So it only takes 5 values. So we have 0, 25, 50, 75, and 100. So 50 means that 2 counts out of those 4 genes are also orthologous. We also have whole genome alignment coverage over here, and then we also have some orthology confidence assessment. You can also manipulate this table, so you can sort this table by clicking on these arrows over here. You can filter this table from this filter section here, or you can export the table by clicking on this Excel icon. So I want to explore this table a bit further with you. So I'm going to select rodents and related species. And then the second table changes to show me the rodent orthologs. And let's search for mouse ortholog. So I'm going to use this filter. I'm going to type mouse, and now we have mouse mucilis over here. And if I bring your focus to the third column, the ortholog column, we can also view sequence alignments. So I'm going to click on view sequence alignments, and we have the option from two ortholog alignments. So we can view protein alignment or we can view cDNA alignment. I'm going to click on protein alignment. Over here now we have the ortholog protein alignment, and this is shown in cluster W format. If you're not sure what the cluster W format means, we can just click on this question mark over here. So let's click on it. And then it gives you a nice legend of what the different things in our sequence means. So when it's a star, it means that the amino acids are identical. If it's a colon, it means that the amino acids are different, but the function is conserved. Dot means amino acids are different, but the function is semi-conserved. And space is when amino acids are different and there is no conservation of function. And then similarly, we also have a key for the cDNA alignments if you've chosen to view cDNA alignments. So let's go back to our page. You can see that we also have these symbols shown in our sequence. You also have the option to download the alignment by clicking on the download homology button. And I think this is all that I wanted to show you in the first demonstration. Now let's go back to our presentation. So before we move on to the exercises, I wanted to talk a bit about the sequence-based resources. So here we have whole genome alignments, but we can distinguish by pairwise or multiple alignments. And these are used to identify highly conserved regions. So we've briefly touched on this in the very beginning. So these are sequences that evolve slowly, which means these are similar across different taxa, as new mutations in those regions tend to be deleterious and are removed by purifying selection, as those regions are likely to be functional and they could be either coding or non-coding. They could also be used to support problematic gene predictions, so we might improve gene prediction based on homology, and they're also used to define syntenic regions, and synteny means conservation of gene order in a chromosome. So pairwise alignment is simply alignment of a pair of species, and we calculate them with two algorithms. We have BLASTZ, and it's the new version of BLASTZ. In Ensembl, you do not calculate your own alignment, you only choose which pre-calculated alignment you would like to display. So we compare humans against everything else, and we also compare model organisms against related species. So for example, mouse and rat and zebrafish, but also agricultural animals and against each other. Then we have pairwise alignments, which are also used to derive synteny between species. So synteny, as I mentioned before, is basically conserved order of aligned homologous genomic blocks between species. And there are different definitions of synteny out there, but in Ensembl, we don't take the orientation of these blocks into account. So for example, I'm showing you a syntenic region between human and chimp. And as you can see that we have these three genomic blocks, and their order is conserved, so we would consider this region syntenic, even though the middle block is inverted, and as we do not take orientation into account. We also calculate multiple alignments. So these are alignments of multiple species, and we use two algorithms for this. Our main algorithm is EPO, which stands for Enredo, Beacon, Orpheus, and these are which are 3 steps in this calculation. So basically, Enredo splits the genome into genomic segments, then Beacon aligns those genetic segments, and Orpheus reconstructs the ancestral sequence. And we use this algorithm to compute multiple alignments for groups of species, including 38 fish, 17 sauropsids, 46 deuterian mammals, 12 primates, and 21 murine species. Then we also have APO extended, which is the extended version of these alignments. And it was previously known as APO low coverage, which allows for assembly of lower quality to be used. So it allows for fragmented assemblies, and it's calculated for the same group of species, but with a higher number of species, as we also incorporate the lower quality assemblies in this calculation. And on top of that, we have 16 pig breeds and other agricultural mammals. EPO is used for fairly close related groups, but we also have Mercator Beacon, which is used for slightly more distantly related groups of species, and we use it to calculate multiple alignments of 65 amniotes, which include mammals, birds, and reptiles. And you can also find more information on our comparative genomics analysis by referring to this publication on the bottom of my slide. Let's go back to the Ensembl Ensembl browser now, and we're going to explore the sequence-based comparative genomics resources, and specifically we're going to have a look at the HOX-D cluster to find alignments and conservation regions between different species. So let's jump out of this presentation, and I'm going to copy this genomic coordinate, and you can find the demonstration on page number 59 of your coursebook. I'm going to go back to the Ensembl homepage. And from here, I'm going to first select my species of interest, and now I'm going to paste my genomic region and click Go. So this takes me to the location tab, and you can see a bunch of HOX-D genes here. In the middle is gene cluster, which is involved in limb development, and it's highly conserved between species. So I'm going to go to configure this page to add data to this view. So this is on the menu on your left. And also on the left-hand side, if I just scroll down a bit, I can find this section that says comparative genomics. So I'm just going to select this. And let's say that we are interested in conserved regions. So let me just, so we already have constrained elements for 90 Eutherian APO extended track already selected, but I'm also going to turn on the conservation score for 90 Eutherian mammals APO extended. So I'm going to click on this box here and turn it on. Once I'm happy with my selection, I'm simply going to save and close this configuration. So let's just wait for this to load. Over here, we can see the constrained elements for 90 Eutherian mammals APO extended. And we can also see the GURP scores for 90 Eutherian mammals APO extended. These are the tracks that we selected. You can find the GURP conservation scores in light pink. The more positive the value, the more evolutionary conserved the base is. And you can see that we have these curved peaks overlapping with genic regions, especially with gene exons. So I will just show you over here. So this alignment overlaps with the gene exons here. And these scores were used to determine the constrained elements, which are shown in these dark pink blocks in the track above. And these blocks indicate regions that are highly conserved and they correspond to the GERP peaks. I hope this makes sense. Now we're going to go back to configure this page. And we're going to explore the pairwise alignments. So let's go back to comparative genomics. And I'm going to click on BLAST-Z and LAST-Z alignment. So let's say that in this case, we're interested in chicken. Sorry, I've already selected this, but we have selected chicken. We've selected chimp, and I've also already selected mouse. Not mouse lemur. Let's turn this off. It's mouse mucilus. Once I'm happy with my species selection, I'm going to save and close. So our tracks have now loaded, and I'm going to scroll to the bottom of this region, and over here we have our individual species comparison. So the pink color indicates alignment, whilst the white color over here are the gaps. And you can see that the best alignment is found between the most closely related species, so that's human and chimp, whereas the mouse has a few gaps over here that are shown in white, whereas the poorest alignment is between chicken and human, which is very fragmented, as you can see. Okay, so there are some questions about configuring this page and for me to repeat it. So what I did was I went to configure this page, and now I wanted to focus on pairwise alignment. So it is from BLAST-Z and LAST-Z alignments. And from this, I chose the species chicken, chimpanzee, and I chose mouse, Mus mucilus. Once I was happy with all of my selections, you can save and close by clicking on this tick button over here. Anamika, can you please check if you specified the species of interest when you pasted your genomic region? Because I think that might be an issue if you haven't specified your species. So once our tracks have loaded, I will scroll down. So this is our region in detail view. I'm going to scroll down in the region of detail view, and these over here, these pink blocks indicate alignment. So we have our individual species comparison over here, and as I mentioned, if it's pink completely, so you can see for chimpanzee we have the best alignment. Then for mouse we have some gaps over here which are indicated by these white blocks, and for chicken we have the poorest alignment between chicken and human, which is kind of fragmented, and there are loads of gaps here. I hope this makes sense now. So this is a graphical view of the alignment, but you can view the alignment between species or groups of species as text as well. So let's go back up. And on the left-hand side menu, we have these options for alignments image or alignments text. So I'm going to click on alignment text. This will not show you any data for now because we haven't selected an alignment. So we're going to select an alignment by clicking on this blue button here. And from here, we can either select our alignments from these selecting multiple or pairwise, so from these menus here, or you can also type in your species that you want to compare with. So I'm going to choose pairwise, and I'm going to choose rodents. From rodents, I'm going to choose rats and mice. And from this, I'm going to select mouse reference. Once I've selected my species, it's also going to appear on the right-hand side. Now, once we're happy with our selection, we can simply click Apply. So over here, we have this table that shows you a summary of alignment blocks. So a region of interest might align to different regions in mouse, and in that case, we get a large alignment block. So we've got a large alignment block on human chromosome 2 against mouse chromosome 2, and then we also have a smaller one on human chromosome 2 and mouse chromosome 7. And since we're after large alignment blocks, as they are more reliable, so I'm going to click on block 1. And over here, we have a list of aligned regions, followed by the sequence alignment over here at the bottom. So I'm going to click on display full alignment. So let's scroll down a bit to view the full alignment. And these red texts over here are the exons, and the alignment in the coding region is much better in the intergenic region. Okay. Now we're going to come back to the left-hand side, and we're going to click on region comparison. So this option allows you to have a more detailed comparison of the region between mouse and human. And for this, we're first going to select our species by clicking on this blue button here. And I'm now going to search by typing in my species. I'm going to select mouse reference. So my selection has appeared on the right-hand side, and now I'm going to click Apply. So this view is a bit like the location tab, but you can see a detailed region in view side by side for two species over here. And we first have the human region. So let me just scroll down a bit. So we first have the human region, and then on the bottom, we have the mouse region, and the aligned regions are represented as these pink blocks over here, and the homologous sequence is highlighted in green. You can further customize this view and add extra stuff by clicking on Configure this page. So let's select Configure this page. And from here, you will find a list of species at the top of the page. And this allows you to configure each species separately. So we can first choose human and configure the tracks, the data tracks, or you can then choose mouse and select the data tracks that you're interested in. Let's go back to our original page. And the last thing that I want to show you is Synteny. This is also on the left-hand side under the comparative genomics section. So let's click on Synteny. And as I mentioned before, that Synteny is used to study chromosome evolution and is only performed for chromosome level assemblies. So on this page, you can see that we have the human chromosome in the middle with mouse chromosomes around it. And a region of interest is shown here in this red box in both species. The large-scale Synteny regions are shown as colored blocks. And you can see that the blocks for human chromosome 2 correspond to different mouse chromosomes. So we have the human chromosome 2 mentioned over here at the top of the image. So for example, this purple box containing a region of interest corresponds to the mouse chromosome 11, whereas this green box over here corresponds to this green box on human chromosome 2 corresponds to mouse chromosome 12. You also have the option to change the species that you're comparing to by selecting Change Species from this drop-down menu here. And you also have the option to change chromosome. And then finally, we also have this table at the bottom, which lists the synthetic genes in our regions of interest in both species. So we have the Homo sapiens, and then we also have the mouse. And this is all that I wanted to show you about comparative genomics. Let's go back to our presentation because it's now time for the exercises. And you can find the exercises on page number 62 of your course book. And if you have any questions or if there is something that wasn't very clear, please don't hesitate to ask us on the living document or either on the chat, and then I'll be happy to go through it again. So we will give about 25 minutes for the exercises. So there's a question about the cluster format. Let me just go back. So you can view the cluster format by going to Alignments Text. And this is on the left-hand side. Actually, no, let's just search for our gene of interest, which is BRCA2, human gene. And then on the gene tab, we're going to go to orthologs. And from this, let's just select African ostrich, for example, and we're going to click on View Sequence Alignment. And then just you can choose either protein alignment or cDNA alignment. I'm going to choose protein alignment. And now we have the cluster W format for multiple sequence alignment. And if you want to find the key for these symbols over here, you can just click on this question mark next to ortholog alignment, which will open a new window. And it will show you what the different conservation codes mean. There's a question about the GURB scores, so let me just show you on the screen. I'll use the bracket 2 example just because I'm already here. So let's go to the location tab. And I'm going to wait for my region in detail view to load. Okay, so over here we have our GURB scores that are shown in light pink. And the more positive the value, the more evolutionary conserved the base is. So you'll also see that these GURB peaks overlap with these genic regions. And they are especially the peaks overlap with the exonic region. So you can see, you can also draw on this region in detail view, and then you can see the overlap of the peaks with the exons. And exons are filled boxes, or boxes. Coding exons are filled, and non-coding exons are non-filled, which are something like this. I hope this helps. So there's also a question about exercise 2, how we can see the 13 out of 46 mammals have an alignment. Okay, let me just see the question. Okay, so this is a cow gene, and this is ABCC11. So we're going to go to the Ensembl homepage, and I'm going to now select my species of interest. I'm going to select cow from the drop-down menu here. And now let's type in ABCC11 and click Go. So this is the ABCC11 cattle gene. And let's go to the location view for this. And from here on the left-hand side, we can choose to view alignments either as image or text. I'm going to choose text. And this is on the left-hand side on the location tab. And now we're not seeing any data yet because we haven't selected an alignment. So let's select an alignment by clicking on this blue button here. And the 46, what does this exercise ask for? So the 46 deuterian mammals APO, this is a multiple alignment, so let's select multiple and select the 46 deuterian mammals APO. Similarly, you can also type in this text box here. So over here on the right-hand side, we have all of the species that are included in this 46 deuterian mammals APO. So let's now click Apply. And over here we have some information about the missing species. So we have 13 species that have no alignment in this region out of the 46 that we chose to see. So I hope this answers your question. How are we doing with the rest of the exercises? We still have about 15 minutes, just under 15 minutes until this module is finished for today. So please do let us know if you have any questions related to the exercises or maybe from the other modules that we've covered previously, and we'll be happy to answer. So there's a question about the cluster format for this. Just click on the biggest block. So I'm going to choose block 1. And then if I scroll down, we have some gene trees and also some information about the alignments, and you can also display the full alignment. But to download it, let's just go back up and click on the download alignment link here. And then you have the option to download in FASTA, ClusterW, and various other formats as well. And you can also guide to file formats from here or preview them. It's now time for us to end today's module on comparative genomics. Tomorrow, we're going to meet again at the same time, and we will be covering Ensembl regulation, and I'll be speaking to you about the Ensembl regulatory build. Thank you very much again for all of your questions, and have a good evening.